Well, welcome everyone to the ICCS VESRI Journal Club, the November edition. It was very exciting today to welcome CALIPSO into the family, and we're going to have a triad of talks today, which is very exciting. So we will start with Philippe Ciais. No, I already said it wrong. Ciais. Ciais.
Yes.
CS, who is a lead PI and is going to give us an introduction to the CALIPSO project. So take us away.
Okay, thank you. So CALIPSO means carbon losses in plants, soil, and ocean. And I think we all know that the increase of CO2 is due to human emissions, but it could be worse actually, because our planet has a carbon cycle that managed so far to absorb, on average, about half of the carbon which is released by emissions. And Corinne Le Quéré and Drs Philippe Ciais and a lot of other people are updating very carefully the carbon budget, which is a balance between what humans put into the atmosphere as land use change and fossil CO2 emissions. And the fate of this carbon is partly to stay in the atmosphere, and this is increasing the greenhouse effect and driving climate change, and also the fraction which is absorbed by land and ocean. And even though the perturbation that you see on the top, the amount of carbon we put every year, has increased tremendously between the 1950s and now, basically it has been multiplied by 5. The response of the carbon cycle, despite its incredible biological, physical, and chemical complexity, has behaved like what you would call in physics a first-order perturbed system. You multiply by 5 the input, and the fraction of carbon, which stays in the air, has remained remarkably proportional to the input. And this is indicated by the airborne fraction, the ratio of the atmospheric CO2 growth rate, aka the fraction of emissions that stays in the atmosphere, divided by the total amount of CO2 emission. And it has some oscillations. Some years, in particular, because of the land, you have a very big weakness of the absorption capacity, so the airborne fraction is high. Some other years, the land in particular is stronger and it captures more CO2, but on average, since we have atmospheric measurements at Mauna Loa, the airborne fraction stays very constant. And if you look at what Earth system models tell you for the future, they are driven by different atmospheric CO2 concentration scenarios. And you look at the left at the evolution of the ocean sink and at the evolution of the land sink. Each color is a scenario. In one scenario, CO2, as you can see on the right, goes up and then it peaks and goes down. It corresponds to a strong mitigation scenario. And in the worst case scenario, the SSP 58.5, CO2 is going up and you have a lot of climate change. And you can see that for the ocean, more or less, there is a kind of proportionality of the ocean sink to the CO2 scenario. But the most warming intensive scenario shows some kind of saturation of the ocean sink because the climate is too warm and slows down the ocean circulation. The ocean gets more stratified and captures less CO2. For the land, it's pretty much the same, but there is a big difference, is that if you look at the uncertainty between the Earth system model, for the ocean, the spread between the models is smaller than the difference between the scenarios. So you can tell to policymakers, if you follow this scenario, this is what you will get. On the other hand, for the land, you can see that the uncertainty related to structure and parameter uncertainty of the land carbon models is so big that sometimes the spread between the models for a given scenario, like the brown one, is larger than the difference, which is quite a problem. Does it mean that we know everything about the ocean? I would argue no. It's just that all the models kind of look the same, and maybe they miss sources of uncertainties that we will explore in CALIPSO, but this is actually the situation. Quite pathetic for land, and maybe, let's say, too good to be true for the small spread of the ocean models. So the question we ask ourselves in CALIPSO is there a risk that the carbon fraction starts to increase, that carbon sinks become unstable? There are a lot of papers about, detecting signs of weakening of this. This is a kind of statistical analysis with the time series, autocorrelation, and various change, and they analyzed more or less observation-based estimate of the carbon fluxes, and in all the red areas, they concluded that you have a sign of both a kind of slowdown of the recovery after the perturbation and an increased variability, which is a sign that the system may approach a transition, but it's quite uncertain. And then you have a lot of regional papers that show that, for instance, in some regions, you have an abrupt increase of extreme fires, that you have increased mortality during dry and hot years in tropical forest, or increased mortality in European forest, and so on and so forth. So the starting point of CALIPSO is that the Earth system model, which are used in IR6, are not bad, but because they are very complex, they take a lot of development, and they are maybe missing some key processes that could destabilize the carbon cycle. Those key processes, you probably have heard about them in the media already. Extreme wildfires can wipe out incredible amount of forest and carbon, and this carbon will be slow to recover. So you have a net increase of CO2 in the atmosphere. Forests die back from severe drought. Also increased losses of soil carbon from microbes that kind of wake up and eat more organic matter and respire more CO2. And this is particularly important in the soil of the Earth that have the largest amount of carbon per unit area, which are in peat and in permafrost. And these are the main things that we want to focus on for the land and for the ocean. There are also important processes that maybe Corinne will mention that lead to destabilization of the kind of biological pump in the ocean. So very quickly, wildfires can respond non-linearly and increase a lot during dry years. It's kind of inevitable because the Arctic and the boreal regions are becoming warmer. Some summer are very dry, and you see, for instance, in 2021, we have a fire anomaly detected from atmospheric carbon monoxide observation, which is something like 5 times, above 1 sigma. And for the first time, we have a takeoff of this boreal fire emissions, which is larger than the tropical fire anomaly that we have seen so far. 2023, you all remember, in Canada, we have also this incredible amount of carbon, which is like today 5 or 6 times the fossil CO2 emission from France, which has been released in the atmosphere and may not recover soon. So in CALIPSO, we want to study and to include extreme fires that are very intense and spread very quickly. What is their effect on the loss of carbon by the terrestrial reservoir? Second point, risk of forest dieback. In terms of carbon, the largest carbon pool in tree biomass is in the tropics. The Amazon alone contains 100 billion tons of carbon. This is a lot. And if you have a mortality of 4%, every year you will lose 4 petagrams of carbon, and that's quite high. So the problem is that detecting mortality in this forest is very elusive. We have very few ground observations. They show that you have different spatial patterns of mortality, and they show that during dry year, like the most recent El Nino, you have an increase of mortality. But typically, during this El Nino, the epicenter of the drought in the Amazon was in this corner of north-central Brazil, and it is not covered by field samples. That's the reason why in CALIPSO, we aim to look at satellite data because they have a very good temporal and spatial coverage. Satellites see optical properties or radar backscatter or LiDAR height, and we want to transform that into mortality. Here you have an idea of the situation. The fact that if you look at remote sensing data, for instance, a kind of new map of biomass from ESA or microwave product that relate to biomass from the SMOS satellite in the middle, if you make the difference between the biomass after a known mortality event on the ground, and before there is a negative value. But in the current carbon models, the same being used for the IPCC, there is no change because, believe it or not, the terrestrial carbon model, all of them, none of them has a parameterization of how drought kill trees. They consider that mortality is a constant factor, so you have a first-order kinetic laws that controls the output of carbon from tree mortality. And obviously, this is not what happens in nature, and this is something we want to simulate in models. Basically, simulate the hydraulics, how water is transported from the soil to the root, to the stem, to the leaf, to the atmosphere. The phenomenon of cavitation or embolism, formation of air bubbles that prevent water flow in the sap, and in the end, kill trees. And we want to calibrate those very complex physiological processes using ground and satellite observations. Okay. Now, for permafrost, as you probably have seen in many media, permafrost are black soil. They are very thick. Some of them are more than 30 meters thick, and they contain a lot of carbon, up to 30 to 40%, more in the case of peat carbon. Simply, most of this carbon is frozen. It's below what is called an active layer. So as long as it is frozen, it can be very old, and it's not dangerous. But what is dangerous is that with the warming of the air, the soil will eventually warm, and it will liberate carbon that is available to microbial decomposition. And we do see in all permafrost warming experiments that there is a very strong release from soil. And this is something we want to study in CALIPSO. The difficulty is to capture the high landscape complexity of the Arctic regions, because in the same grid cell of your model, like 1 degree by 1 degree, you can have a bit of peatland, you can have some thermokarst lakes, you can have some upland frozen tundra with a lot of ground ice in the soil, you can have some forest, and all of these landforms have very different carbon content and very different responses to warming. So one of the challenges is to use data and to increase the complexity of the hydrology and the soil thermics in the models to be able to capture not only the gradual warming of the soil that more or less all models have, but the phenomenon of abrupt permafrost warming. And this has a name, it is called thermokarst. So when you have thermokarst, it creates a liberation of carbon. And this is completely absent for the current Earth system model. This is an example. In Russia, for instance, our colleague Sergei Zimov has been measuring the active layer depth, very basic. He has a stick that he put in the ground. And before an abrupt thaw event, the kind of permafrost layer which blocks the pulling down the stick was something at 40 centimeters. And after a very warm year, you can essentially pull a stick of 3 meters in the ground, which means that at this particular location, all the permafrost is capable to disappear in simply 1 year. Then for the ocean, well, the ocean is a kind of the ocean biology is extremely complex. You have much more trophic layers than on land. And what is primordial for the sequestration of carbon is the relation between the CO2 removal in the sunlit layer of the surface ocean versus the CO2 production by mineralization, also microbial decomposition, like in soil, below the surface layer. And this depends on the quality and also on the density of the dead plankton particles. If they are very heavy and very big, they will sink to the bottom of the ocean, and it will be really a kind of carbon removal. If they are very puffy, very light, very easy to eat for the microbes, they will be basically recycled back as CO2 in the atmosphere. And the ocean group will look at two processes that are susceptible to change this ratio between uptake of CO2 by photosynthesis and mineralization by the decomposition of this kind of marine snow below the ocean surface or within the ocean surface. One is the existence of virus. Virus can really explode cells and they make them more puffy, so much more kind of easy for them to release CO2 back to the atmosphere. And also changes in the zooplankton diversity, because zooplankton maintain, like cows, a grazing pressure on the algae, and any small change in this plankton diversity is capable to lead to a very strong change in the net CO2 fluxes. And well, would not believe maybe that virus is an important factor, but we have millions of subsea camera images here taken from the Tara ship. And these cameras allow to assess and to count the species type, and also to look at the presence of viruses. And they found a very strong relationship between the presence of viral groups in the kind of sparse images and the ocean carbon export. Carbon export is basically controlling how much ocean can take up CO2 from the atmosphere. This process is absent from the current ocean biological model. It's very challenging, but again, we have this unique asset of subsea cameras. I think it's several million of images, helped with machine learning and image recognition patterns tools. We can basically classify the different type of species and try to have a new generation of ocean biological model that will couple, of course, the ocean physics with these new processes related to the ocean biology. So in a nutshell, we hope in CALIPSO to make, we say the giant leap, but I found it too arrogant, so maybe a major improvement in the representation of carbon loss processes. So we focus on loss processes in Earth system models related to what we believe are three important or critical knowledge gaps in the global carbon cycle: potential factors that could drive losses of tree biomass from drought events and fires, increased soil carbon decomposition. I spoke about permafrost, but also changes of microbial diversity, just like in zooplankton diversity in the ocean, has also potentially the potential to change or to accelerate the decomposition of carbon in the soil and destabilization of marine biota. So that's what we want to do. Of course, we will try to develop those new processes in standalone version of the land and the ocean model. And then the goal is to integrate all those processes into at least one fully coupled Earth system model, the IPSL Earth system model. It's important to think about the interaction in advance, because all these processes can be more important through the interaction they have with other processes than by themselves. For instance, fire will alter the productivity and the recovery of forest, not only the emission when you have a mega fire. So this last work package of CALIPSO is coordinated by Pierre Friedrichstein. And the plan is to produce during the first 3 years a number of development, to calibrate the development, to use data assimilation, parameter inversion scheme, and all the new observations that we will collect in the project. And then to start the integration, of course, before, in order to have a better idea of the importance of those destabilization processes for a range of future climate scenarios. So the team, I think, no, I don't have, sorry, a presentation of the teams, but we have about 10 teams. It's a very international project. The teams are located in Europe, in the United States, in China, and in Australia. They are all experts in their field. And well, we work together because some of the developments we want to achieve are, I think, impossible or would be extremely difficult to achieve by a single research group. So we will put in common our knowledge and our resources, for instance, knowledge in soil microbiology, in very detailed soil microbial models, and then in land surface model to try to achieve the goals of the CALIPSO project. I think I will stop there, and feel free to ask if you have any question or comment.
Right, thank you so much for that. I suggest that we move on to Corinne's part of the session now. Corinne?
Yes, thank you. Yeah, absolutely.
Great.
Thank you very
so if
much.
you pop that up, your first slide up, and I will introduce you.
Thank you.
Okay, so we can then move on to the ocean challenge part. So Corinne Le Quéré is going to talk about testing machine learning-based reconstructions of particulate matter in the ocean. Thank you, Corinne.
Thank you, and thanks everyone for being here today. So in the 10 minutes or so for my presentation, I want to discuss some work that I did, mostly a postdoc working with me, Anna Sommer, did with me, and which we published in the Geoscientific Model Development Journal earlier this year on testing machine learning-based reconstruction of particle matter in the ocean. So just a little bit of background. Philippe showed this figure already. I mean, there's really one crucial process that is ecosystem related in the ocean, and it's how much organic matter sinks out of the ocean and into the deep ocean, because this process really acts to isolate carbon from the atmosphere. And if we didn't have it, the atmospheric concentration of CO2 would be 200 parts per million higher than it is today, which is equivalent to about 2 degrees. So it's an extremely important process. It's a process that is limited by nutrients, so not directly limited by CO2 level, but it does respond to climate and climate change in ways that we really don't understand how it does. So we're in the process as a community of building ecosystem models that include more realistic and fine representation of some ecosystem processes. In CALIPSO, we have decided to focus on the very small ones, which are the viruses, and on the very big ones, which are the zooplankton. But this builds on quite a lot of work and on knowledge that some of the challenges that we have in the community is to understand the linkages between the community, the plankton community structure at the surface, which plays a fundamental role in determining the morphology, stickiness, density of those particles, organic particles that are created at the surface, and hence their sinking speed out of the surface layer where they become isolated from the atmosphere. In parallel to like the theoretical understanding and the model development, there has been some pretty incredible developments in observations, and in particular in the underwater video profilers. There's nearly 150 million images available today, just camera images of the stuff that is in the ocean. And what you see at the bottom here is actually a machine learning characterization of these particles into dark matter, essentially elongated matter, which is mainly fecal pellets from zooplankton, flakes, which tend to be aggregated, small particles, fluffy particles, or agglomerated big sort of particles that can or not sink rapidly to the deep ocean. And a big question that we've been asking ourselves is, how can we use these observations to inform model developments? And the observations are relatively new. They give us 3 variables. They give the number of particles per volume, they give the morphology of those particles, and they give the gray level of the particle. If you park this information for a moment and look at the kind of models that we're developing and that we'll be using in CALIPSO, the one you have in front of you is the one that I developed in my group, the plankton model. It includes the representation of 12 what we call plankton functional types. So there are types of phytoplankton and zooplankton and bacteria that are regrouped into groups that have similar impacts on the carbon cycle in the ocean. So we have 12 of those. the zooplankton eat the phytoplankton. In the ocean, recycling is extremely fast. Everything gets eaten in the ocean. There's barely any standing stock of organic carbon. And then the resultant of all of these processes, so one eating another, the small one the bigger one eating the smaller ones, and so on, results into this sort of marine snow and organic particles that sink to the deep ocean. And the underwater camera is really the amalgam, so the sum of all of what has been sinking in the ocean. So with Anna Summer, what we did is we thought, can we, in fact, use the underwater camera information to nudge the production of particles in our ocean models based on knowledge, inferred knowledge of the relationship between the surface ecosystems and the particles? And so we wanted to sort of introduce a new way of thinking about process models by not having them only rely on understanding of processes and parameter tuning, but actually actively nudge them towards the production of observed number and sort of characteristics of particles. And you'll see that the approach is not as promising as we were hoping for, but still the exercise has been interesting. And so I'll run you through the results here. What we have done is, so on the left-hand side is the distribution of the vertical profiles of the underwater camera that are co-located with surface ecosystem and vertical ecosystem distribution from the Tara Oceans cruises. And so we've used most of the data to run a machine learning model. And we have also sampled testing data outside of the locations where we did have complete observations. Now, we haven't applied this method to the actual observations themselves. What we have done instead is produced a synthetic data from the Plankton 12 model, because we knew there what the particle distribution was, and we had full information about the ecosystem distribution. So we sampled the model and we got sort of a perfect test bed to try out our thinking on this. We tried two machine learning methods, the random forest and the RG-boost method. They are distinguished in the sense that they are RG-boost has an iterative process. So it first ingests some of the data, then learns, and then ingests some more. We've done 27 different sets of drivers to reconstruct our two particles in the model, the small one, POC, and the large one, GOC. And essentially, the 27 driver sets are a function of, the first one has no plankton functional type information, so no ecosystem information at all, so just nutrient, temperature, and physical data. The second sort of set has PFT, so all the PFTs, then it has either surface or depth integrated PFTs. The third set has various combinations of PFTs, mostly regrouping the big ones together and the small ones together. And the last set has time dependence, so that the machine learning model was able to access what happened the previous month. And so the results produced obviously a lot of statistics. We looked at the RMSE error, we looked at the absolute bias, and we looked at the correlation. And you have on the left-hand side, the big one is the small particles, and on the right-hand side, the big particles, training data, validation data, and test data. So just as basic, you know, of course, the machine learning algorithm does better in the training data, a little bit less good in the validation. So RMSE increases validation and test data. For those, that's all expected. Absolute bias the same, and correlation is a little bit deteriorated. That's the same. But in terms of substance, what comes out of this is that generally the small particles are better reproduced than the big particles. But the most interesting for us was that the PFT information, so information about on the ecosystem, when that was introduced, you really did see an improvement in the reconstruction of the data with the PFTs. And then when we included too much information, which is the specific vertical profile of all the PFTs, which is this sort of middle box here, you start to see a more deterioration because the model tended to be overfitting the observation. So this allowed also us to say how much the PFT improved the fit to the observations or the reconstruction, and also to see what was the most important drivers for the spatial variability of the small particles and of the big particles. And you see that in both cases, the microzooplankton is a very important organism, and this is actually also what we think from the reality, in addition to some of the physical variables. So overall, the reproduction looks physically, visibly like this. So the small particles on the left-hand side, the top of the plankton 12, and the best reproduction we have at the bottom. And on the right-hand side, the big particles. And there are regions where there were no observations, and this is where the fit is less good, but overall you could reconstruct a global map for that. So our conclusions was that you could in fact reconstruct the map. The PFT information improves the reconstruction. That information about microzooplankton was particularly interesting, and technically the RGBoost performed better due to the iterative nature of the algorithm. But overall, we felt that the results were in fact too dependent on the setup and on the actual decisions we took for the drivers of information to, and we thought that this would not in fact improve the existing formulation that we have for the particulate dynamics in the model, and we decided not to pursue this part technically and introduce this in the model, and rather stay with our more conventional approach for now, which includes process-based model developments and validation with the particulate data afterwards. Thank you very much.
With sort of crawling back onto the land, I think, with Daniel Goll, who's going to be talking about machine learning for accelerating model spin-up.
Thanks. You can hear me?
Yep.
So today I will talk about a practical example of how we want to apply machine learning in the CALIPSO project, here specifically for accelerating the computational performance of our land surface models. And this work has been done by Jan Sun in a PhD thesis, and it also has been published and also commented on in Global Change Biology. I think this was this year or last year. I can remove this thing maybe. So I guess you all know we live in a world where we have ever-increasing observational data sets, also known as big data, and we have complex mathematical models which resolve hundreds of processes, so-called big models. And these big models, their computational efficiency is decreasing more and more, while we don't have the same pace with increasing computational capacities. So we have a problem in integrating this large wealth of observations into our process-based models, because they are just too slow to integrate the data. So this is the main problem. And for our land surface model, which all include geochemical cycles like the carbon cycle Philippe introduced, they require a model spin-up, which means we have to equilibrate the biogeochemical cycles to the boundary condition of the initial state of our model experiments. That's most often, in our case, before the human disturbance of the carbon cycle, so pre-industrial conditions. And this spin-up can take up to 89% of the computational demand of a model experiment, because it takes many, many years to equilibrate a model which couples processes which operate on a time scale of minutes, like microbes, to more slowly processes which operate more on a time scale of hundreds to thousands of years, like if you think of solid organic matter or permafrost. So we tackle this computational bottleneck of the model spin-up. But when we started this work, already tools existed which accelerated the model spin-up, but they all have major shortcomings which we aim to overcome with our tools. So these shortcomings are, on the one hand, they rely on linearity, and when we introduce, for example, microbes and how they respond with permafrost and the carbon in the soil in these high latitude regions, we increase the nonlinearity in these models. And on top of them, this is often based on semi-analytically solving equations, a set of equations, and this requires quite some thinking to adapt these tools whenever you change the structure of your model, which we do constantly. So these tools are very hard to maintain, in particular in our community where we don't have these mathematical skills constantly at hand. So we wanted to come up with something which overcomes these shortcomings, and we exploit the fact that in the current generation of land surface model, each of the model pixels is perfectly independent of all the other pixels. So there is no communication between pixels. So our idea is to only equilibrate a subset of pixels and then use a machine learning algorithm to predict the remaining pixels. And as each pixel and the computational demand of such a model that scales with the number of pixels simulated, so the less pixels we brute force spin up with the land surface model, the lower the computational demand. To do so, we made a machine learning based setup, which is based rather on pragmatic choices. We just wanted to have a proof of concept that this can work. So what we have been doing, we take the input forcing of the land surface model, which are global maps of climatic variables like temperature, precipitation, wind speed, wear, plant functional types, etc. So we have a couple of these maps. And we use the K-mean clustering procedure to select pixels which are representative for the whole globe. And then we selected these pixels and we run brute force equilibration simulations on this subset of pixels. We choose about 15-16%. And then we also tweaked a bit the training data set by doing a synthetic minority oversampling technique. And we trained a tree-based bagging ensemble and used then our global land surface model input fields to do the global extrapolation of the steady state of all state variables which define the biogeochemical cycles in a land surface model, here at the example of orchidea. So the question is, how well can we actually predict the equilibrium state of the models which the model pixel we do not brute force equilibrate? To test this, we computed basic statistical indices like R-square, normalized root mean square error, and the slope between the machine learning predicted steady state and the true steady state from simulation we did where we brute force equilibrated all pixels. And you can see here one of the plots we produce. You have on the y-axis a different plant functional type. So we grow an orchidea on every pixel 15 different ecosystems, from tropical trees to boreal needle leaf trees. And on the x-axis, you see the different state variables of the land surface model. Here as an example of the soil organic carbon pools. We have 3 of them. And here you see the R-square. We did the same not only for the soil organic carbon, but also for biomass carbon and litter carbon. We also did the same for nitrogen and phosphorus pools, but I don't show it here. And you can see that we get rather well correlations in these different compartments. Some pools don't work so well, some PFTs don't work so well, but luckily these pools which equilibrate rather fast. For example, we have the labor carbon reserve in trees. That's a short-term carbohydrate storage within the sap flow of trees, and we can equilibrate that with an additional Orchidea simulations, which we restart from the machine learning-based equilibrated state, and we only run a few model years to equilibrate these pools. However, still you see there's some issues with the machine learning predictions. So we were testing to what extent these biases in the equilibrated state affect the typical land surface model application. And here we choose a very famous application, which is land surface model simulations, which contributing to the global carbon budget updates, which happen each year. They were led by Corinne, and now they're led by Pierre. And you can see on the right-hand side a typical picture, which is based on this land surface model simulating the land carbon balance. So we did a test with different versions of Orchidea at different spatial resolutions, where we run this kind of historic simulation, restarting from a brute force global equilibrated state, and another simulation where we used our machine learning accelerated spin-up tool. And this is shown here. You see here in colors lined different versions of Orchidea. So we have Orchidea version 2, we have a nutrient-enabled version 1.2 and 1.3. And in the gray broken lines, you see the same simulation, but restarting from a machine learning accelerated spin-up. And we have not only the global land carbon sink on the top left, but we also looked at different parts of the Earth, like Northern Hemisphere, Southern Hemisphere, and the tropical regions. And we find very, very minor biases in this typical prediction of a land surface model. So we think this approach is very promising, and we can use it without having much alteration of our model predictions. So how much time do we save? I have here a plot which shows the computational demand of a model experiments using the machine learning accelerated spin-up relative to traditional spin-up. And you can see here in this colored bar, bluish, gray, greenish colors, we only need about 20% of the time because we only simulate roughly 20% of global pixels, and the other 80% of the pixels we predict by machine learning, and the machine learning tool itself has a very small time consumption, which is negligible compared to the time consumption of the land surface model simulations. So to conclude, with our tool, we think a time saving of 1 order of magnitude is in reach. It's agnostic to gridded models, so it's beyond terrestrial biosphere model or land surface model, so it's applicable to all models where we have pixels which are independent from each other. And it can also be combined with existing spin-up acceleration procedures, which we also showed in a paper, to even further reduce the time consumptions. And this is really needed because it opens the door to a wide variety of model applications where complex nonlinear models benefit most from this tool. I don't know if I have time, maybe quickly. What comes next? So our ideas how to proceed next, also in collaboration with the ICCS. First, we want to improve the software quality for the end user and then publicly release it on GitHub. We want to adapt it, or we currently adapt it to the CMAP 7 version of Orchidea. We're discussing if we can adapt it to other land surface model used within the CALIPSO project, and we also want to exchange the machine learning algorithm with something better suited to the task, and also optimize the parameters. And in collaboration with other groups, we wanted to add new functionalities, for example, using the tool to go from a rather low resolution to a high resolution, which is very useful for really applied scientific information to farmers, for example. And we're investigating if we can replace Orchidea within the calibration procedure of the coupled Earth system model, where you have an ocean model, atmosphere model, and a land surface model, which in the case would be Orchidea. So this is something we'll be working on within CALIPSO, but also beyond. And with this, thanks for the attention, and I'm happy to take questions.